Hey AP Bio, this is video 1 in the statistical analysis of the effect of lectin on mitosis in onion root. This video is only going to focus on how we test hypotheses. So, what we will do today, let me move my picture down. We're going to define what a null hypothesis is because that's new for most of us, because what we've been making throughout all of our time that we've learned science is alternative hypotheses or working hypotheses. And we're going to see how those are different from a null hypothesis. And the main reason that we make a null hypothesis is so that we can actually run a mathematical test on it. And then we're also going to discuss why we use mathematics and statistics to make biology meaningful. So, let's look at these two different types of hypotheses. And then down at the bottom, you see the little cartoon, and the question is, what's happening? And all of these people are giving null hypotheses. Absolutely nothing, no difference, no change, no effect. And that's what a null hypothesis is. A null hypothesis says that there's no cause and effect relationship between an independent and a dependent variable at all. What we normally make are what we call either working or alternative hypotheses. So it's a alternative to the null. So an alternative hypothesis or a working hypothesis, normally we make some type of observation, we see some type of phenomenon, and we make some tentative explanation that explains why that happens. That's what we normally do, and that's called our working or our alternative hypothesis. The main thing that is important is because we cannot ever get absolute proof. We can never be 100% confident in anything. And because of that, we use statistical hypothesis testing that allows us to reject the null, because I can reject a hypothesis, but I can't ever prove one. And we're going to talk about why that is the case. So, what we have here, here's an example of an experiment. We have this teacher, Mr. Stewart, and he has his classes conducting a mark and recapture investigation with snails in a stream. So in a mark and recapture investigation, you would take snails up, you would do some type of marking like fingernail polish or something on their shells, and then you would put them back exactly where you found them, and then you would come back later and capture them again and see if you have recaptured any of those snails in that same area or not. So the purpose of this investigation, the question that they were looking at was like, what type of environment do snails prefer? Do they like upstream or do they like downstream? So they actually did this experiment in two different types of stream beds, one that kind of is sandy and the other one that has a more rocky bottom. So a working or an alternative hypothesis for this investigation could be snails favor whatever environmental conditions are upstream. And I think snails are going to move upstream in that direction. Probably like there might be a higher dissolved oxygen content upstream. The null hypothesis or the no effect would be that snails show no preference in upstream or downstream. They're going to be pretty much relatively equally distributed in the stream overall. So they're not going to care if they're upstream or downstream. They have no preference. That's always going to be our null hypothesis. No relationship, no cause and effect. So what we're going to be doing is throughout this week, we're going to test these null hypotheses. So if we can do a statistical test and we can reject that null hypothesis, then we know that there's something going on, some type of effect, right? We don't know the cause, but we know that there is an effect. So when we do this and we do all these statistical tests, remember, your null hypothesis can be rejected. And if it is rejected, that does not necessarily mean that your working hypothesis is true. We can't prove it, but it does support it. So where your alternative or your working hypothesis can only be supported if we reject the null. So let's look at an example of that. Let's say that Mr. Stewart's class went through, they collected all these snails, they collected all of their data, and after they did their statistical test, they rejected the null hypothesis. They said, you know what, that's wrong. There is an effect here. Snails are showing some type of preference or something going on. That would support the working hypothesis that snails might favor upstream and move in that direction, but it doesn't prove it, because just because I have data that's correlated doesn't mean it's a cause. So there could be other reasons that snails could prefer a certain environment. If I reject the null hypothesis, I don't know, maybe the snails prefer downstream actually and not upstream. That's a possibility. Or maybe they only preferred upstream in one of those conditions. Maybe they preferred upstream in the sandy area but not the rocky. Or maybe they're moving upstream because they can hide under the rocks. There might be lots of other reasons that are the cause of that preference that might not just be because they're upstream or downstream. So here are some other graphs that show a correlation. Two variables that tend to show a linear relationship, but that doesn't necessarily mean that one causes the other. So here's one right here. This one's looking at the number of people who died by becoming tangled in their bed sheets, and we have that correlated with cheese consumption. We know that eating cheese does not cause you to become tangled in your bed sheets and suffocate yourself. Like, that has nothing to do with it. But those data points are correlated, but it's not a cause. Here's another graph. This looks at the number of motorcycle riders killed in non-collision transport accidents, and it's correlating that to the per capita consumption of sour cream. Now, that's just like ridiculous, right? The amount of sour cream has no effect on how many people are going to die on a motorcycle. But you can see that these graphs are kind of aligned, so they're correlated, but this is not a cause. This one's my favorite. Films where Nicolas Cage appeared is correlated with the number of people who drowned in their pools. I know not every Nicolas Cage movie is the best, but it does not cause you to want to drown yourself in the pool. And then here's our last one, the number of letters in a spelling bee, and that's correlated with the number of people killed by venomous spiders. That is a correlation. Those data points tend to align with one another, but this is not a cause. So let's look at the phenomenon that we're going to be investigating. As you can see over to the right, there's a image of Vidalia onions that are growing in a farm field. In an effort to produce higher crops of onions, scientists do investigate how different environmental stimuli, and in this case are going to use naturally occurring chemicals, might increase the roots of onions. Now remember, the roots of an onion, that's where water is absorbed, but also nutrients. So the thought process is that if scientists could find a chemical that could induce root growth, where roots could grow more quickly, that might aid the growth of the overall onion, because normally onion roots tend to be small, and if roots were bigger, they could absorb more water to do more photosynthesis, and they could absorb more nutrients from the soil. So, what scientists have seen in other fields is these certain chemicals called lectins. Lectins have affected the roots of organisms like soybeans. So, here we have scientists reporting that you can have a fungal or a fungus, and that's a pathogen, that affects the growth of soybeans. Soybeans' growth is actually decreased when we have high rainfall. The reason for that is that those roots tend to grow very quickly, but they're poorly developed. Sometimes if you grow too fast, you don't have strong roots, you have weak roots. So these poorly developed roots were always going to be surrounded by this fungus that they noticed. And that fungal pathogen grew in that soil by these roots, and it made this lectin protein that was secreted by the fungus. So what they noticed was that lectin accelerated mitosis. It increased the cell cycle in roots of those soybeans. And let me move over a little bit, and you can see that number 2 right here, this is showing proper growth of roots. But over here, like by 4, you can see that these roots had been exposed to lectins. These roots, notice they're very thick. However, they're weakened roots. They grew too fast. They could not support the plant, and these soybeans actually did die. But the thought process was that perhaps in onion roots, these, a small amount of these lectins could actually be advantageous to the plant, and it could grow the roots at a rate that might actually help the onion, even though it did not help the soybean. So here's our investigation. We're going to look to see if whether the lectin chemical that's created by that fungus will affect the number of cells undergoing mitosis in onions. So what I have for you in table 1 is the actual data of onion tip cells, and we're looking at how many of those cells are in mitosis and how many of those cells are in interphase. That occurred after lectin was added to the soil of those onions. So let me move my face again. Here's the root of the onion. These would be the onion root that had been exposed to lectins as it grew. And then here is the slide, so you can look at a microscopic image of the onion tip. We'll be looking down here at these onion tips. This is at a 40x magnification. When we actually look at those cells, we'll look at it much more closely at 400 magnification, but we'll be able to identify which cells are in mitosis and which cells are in interphase. I'm going to give you the experimental data from the treatment group. So now you're ready to stop this first video, and you're able to answer these first questions in the lab. In this lectin experiment on these onions, what is the null hypothesis? What is the working or the alternative hypothesis? Then I want you to identify the dependent variable. What's going to be measured? What did we collect data on? And explain how that data was measured. What we did not have, remember, I'm only showing you the treatment group. What would happen to the control onions? So tell me about that control treatment. And then explain to me why statistical analysis of our data is going to be necessary to test our hypothesis. Thanks, guys. Please enter these or answer these 5 questions, and once that's done, you'll be ready for video 2.